virulence factors. Hello, my name is James Hayes. Have you ever wondered how pathogens cause disease? They use molecules called virulence factors. Virulence factors are molecules that are produced, secreted, or expressed by pathogens. Pathogens include bacteria, viruses, fungi, and protozoa. Virulence factors contribute to the virulence of a pathogen. Virulence is a pathogen's ability to cause disease. Virulence factors also contribute to the survival of pathogens. There are many different types of virulence factors, and they carry out a variety of functions relating to the infection and colonization of host tissues and cells. One type of virulence factors are toxins. Toxins are poisons for host cells created by pathogens. Another type of virulence factors are adhesion factors. These factors allow pathogens to attach or to adhere to host cells. Another type of virulence factor are invasion factors. These allow pathogens to enter the host cells. Two more types of virulence factors are immunosuppression and immunoevasion factors. These factors help pathogens avoid or inhibit the host immune system response. Let's go into more depth about toxins. Like I just said, toxins are produced by pathogens and they're meant to kill host cells. They kill host cells by inhibiting cellular processes and disrupting homeostasis in these cells. One example of a toxin would be the tetanus toxin. This toxin causes tetanus. It causes this disease by stopping some neurons in the spinal cord from working. Another example of a toxin would be enterotoxin. This toxin is produced by the bacteria E. coli. It causes oversecretion of water and electrolytes. This deprives host cells of these nutrients. Adhesion factors are also another important type of virulence factor. Adhesion factors allow pathogens to attach to the surface of host cells. They help with the colonization of host tissues. Adhesion factors often attach to receptors on host cells. This is an example of specific adherence. Specific adherence involves complementary receptors and molecules. One of these is on the surface of the pathogen, and the other is on the surface of the host cell. Non-specific adherence involves non-specific attractive forces, like electricity, or, say, lipids being hydrophobic or hydrophilic. One example of adhesion factor is the molecule adhesin. This molecule binds to the surface receptors on host cells. This is an example of specific adherence. Another example of an adhesion factor are fimbriae. These are filamentous proteins found on the surface of bacterium. They act similarly to adhesin. This is another example of specific adherence. One more example of adhesion factors is the capsule of bacterium. This is a polysaccharide layer of the cell envelope, often called the slime layer. This is an example of non-specific adherence. Another type of virulence factors are invasion factors. These virulence factors let pathogens enter host cells. Like adhesion factors, invasion factors help with colonization of host tissues. Invasion factors allow pathogens to enter host cells to carry out a variety of processes like killing the host cell, taking its nutrients, among other things. Invasion factors are usually enzymes that break down structural components of host cells. One example of an invasion factor is hyaluronidase, which breaks down connective tissues. Another example of an invasion factor is collagenase, which breaks down collagen. Pathogens also use virulence factors to combat the host immune system response after initial infection. These virulence factors are called immunoevasion or immunosuppression factors. These factors help the pathogen prevent themselves from being killed by the host immune system response. They can do this in a variety of ways. Two of them are not allowing for phagocytosis and not allowing for antibodies to attach to them, among other things. Phagocytosis is the process of engulfing and digesting pathogens. One example of immunoevasion or immunosuppression factor would be the capsules on bacteria. They prevent phagocytosis by hiding antigens, not allowing for the phagocytes to recognize them as pathogens. Another example of one of these factors would be the structural properties of cell walls of some pathogens. These cell walls are resistant to digestion and bactericidal agents. One more